Yes. Okay, so we're setting up the photosynthesis lab, but our kit did not come with a hole puncher. So we actually took a pen apart and we're just using part of the pen to punch these holes. If you had a straw, that would work too, but I couldn't find a straw. So make a bunch while you're standing here, get a bunch to work with, and there you have it. Alright, and still doing the photosynthesis lab, we got our little scale set up, and we pressed 0 to tare it. Oh, that turned it off. And the other thing to note is, can you see this tiny little G up in the corner? This M is for mode, and if you press the mode, it will give you different units, but we want to work in grams right now. According to the lab, you are going to weigh your baking soda with this. So now you're instructed to use a piece of paper, and this is mostly to keep your scale clean, but see, the paper weighs 0.4 grams, so you have to tare it again when you put the paper on. Okay, and you can see this is an exceedingly tiny amount of baking soda, very small amount. If you don't have baking soda, just ask someone. You don't have to buy a whole one.
Exactly 200 milliliters of distilled water into the bicarbonate cup.
Okay, so now we've got our experimental setup. We've got 2 cups of distilled water and 2 of bicarbonate solution. We're testing 2 different things here. We are adding carbon, so that's one treatment of the experiment. And the bicarbonate solution becomes carbonic acid, which is a carbon source for photosynthesis. The distilled water does not have that. It represents the control. But we added soap, a drop of soap to each solution, so that we could break down the waxy cuticle and make it easier for the carbonic acid to move into the plant structure. Now we have our solution set up. We are going to prep up our biological material. We're putting our hole punches into the syringe, and then we're going to suck up 3 mL of the distilled water solution. We're going to work on creating a pressure gradient inside the syringe. And so, hold on one sec, we'll get this set up. Okay, so we're going to push the syringe in until we have just a tiny air space, because we're eventually going to try to get all the air out. And then we're going to tip it into one of the distilled cups and draw up just 3 mL roughly of liquid by pulling the syringe top.
As such.
Okay, and now we're going to kind of tap it, tap-tap-a-roo. We can flip it upside down because we want there to be no air. We want to be able to create a pressure deficit in this tube. So yeah, keep pushing it till the water's at the top. Okay, that looks perfect. Now you're going to put your finger over the top to create a seal. And now if we draw the syringe down, we are creating a pressure deficit in there, just ever so slightly. And we're going to tap it a little bit. We want these discs to fall all to the bottom. After messing around, we decided 3 mL wasn't quite enough liquid, and also more is shown in the picture in the lab report. So we added some more. Now we're at like 6 or 7 mL. And we'll do our pressure deficit thing one more time. If you can imagine, it's like drawing the dish soap in through the waxy cuticle. So we're kind of like opening the doors of the plant cell.
Carbon is by far the most effective.
Yeah, so okay, we finished our photosynthesis lab. So these were our dark chambers, and no spinach leaves are floating in the 2 cups that were in the dark, neither the distilled water or the bicarbonate solution. But then what we see here is on the left you have distilled water only, and on the right you have bicarbonate solution. So we had it under a pretty intense grow light. So the one with no carbon in it, we still saw some spinach leaves floating to the top, but that could have been because they already had carbon dioxide collected inside of them before we started the experiment. But then see here on the right, the one with the bicarbonate solution, all of the spinach leaves are floating up. So this is the result that we would expect to see. Now I'm going to see if I can show you why they are floating up. Okay, so you can see at the rim of this spinach leaf, there's a whole bunch of bubbles. I'm not talking about the bubbles on the top of the water. That's just from us moving the cups. But on the edge of the leaf, you can see little bubbles all around it. And that's the oxygen release from the photosynthetic process. So oxygen is a byproduct. When the oxygen is released, actually those little air bubbles act like rafts, and they are just lifting the spinach leaves up.
The strangers.
Okay, and here's a couple more. You can see the bubbles. They're all over the leaf surface.
He was like, "This is my home." Who is this?